We all have studied the central dogma of gene expression, where DNA is transcribed into RNA, which is further translated into proteins. Let us have a look at translation. Translation is the last step in gene expression and is a consequential step to transcription of DNA to RNA. It occurs in the ribosome of the cytoplasm, during which the coding sequence of mRNA is translated into the amino acid sequence of a protein. It entails three major phases: initiation, elongation, and termination. In this video, we are going to focus on initiation, the first stage of translation in prokaryotes. The cellular constituents involved in initiation step of translation are mRNA strand, initiation factors 1, 2, and 3, and 70S ribosome unit. mRNA or messenger RNA. mRNA is synthesized by the process of transcription. It is a nucleotide sequence present in triplets called codons. There are a total of 64 codons which code for various amino acids, including one start codon and 3 stop codons. The mRNA sequence is always read from 5' end to 3' end and is made up of the nucleotides adenine, guanine, cytosine, and uracil. Initiation factors are undoubtedly the factors that are required for initiation process to begin. These are GTP or GTPase bound proteins. Types of initiation factors. Initiation factor 1 will bind to the 30S subunit at A site. Initiation factor 2 is a GTPase bound protein which also has charged N-formyl methionine attached to it. It also has the anticodon for the start codon AUG on mRNA. Hence, it binds to the 30S subunit due to start codon at P site. Initiation factor 3 binds to the 30S subunit at E site. 70S ribosome unit or rRNA. The 70S ribosome unit consists of two subunits. The larger 50S rRNA subunit, which is made up of 23S rRNA and 33 proteins. The smaller subunit, called 30S rRNA, is made up of 16S rRNA and 21 proteins. On the 30S smaller subunit, there are three sites. A. Acceptor site, which accepts amino acids brought by tRNA. P. Peptidyl transferase site, where the synthesis of polypeptide chain occurs. And E. The exit site, from where the empty tRNA moves out. It starts when the 30th subunit of ribosome recognizes the start codon AUG on mRNA and binds to it. Next, initiation factor 3 binds to the 30S subunit at E site, hence not allowing 50S and 30S subunits to bind together. Then, initiation factor 1 binds to the acceptor site on 30S subunit, hence not allowing any other amino acid tRNAs to bind at A site, thus saving place for elongation factor TU. Now, initiation factor 2 along with charged N-formyl methionine binds to start codon AUG at P site because it has the anticodon for the start codon on mRNA. Binding of all the factors to the 30S ribosome leads to the formation of 30S initiation complex. Binding of initiation factor 2 causes conformational changes in 30S ribosomal unit. This causes release of initiation factor 3. Once initiation factor 3 is released, 50S subunit binds to 30S subunit along with mRNA. Further, initiation factor 2 hydrolyzes the GTP of initiation factor 1, due to which initiation factor 1 is also released. Finally, initiation factor 2 is released as well, leaving N-formyl methionine at the start codon at P site. 70S ribosome on the mRNA along with N-formylmethionine at P site is called the 70S initiation complex. Formation of 70S initiation complex marks the end of stage 1 initiation of translation in prokaryotes. Thank you.